friend, today I am going to tell you about the immunoglobulins. Immunoglobulins are also known as antibodies. When these antibodies are produced after the invasion of foreign material, and the foreign material is known as antigen. As the name suggests, antigen antibody generator. Okay, so what are the antigen may be virus, maybe microbes, or microbial products such as microbial toxins. So B cell produces immunoglobulins or antibodies. The rate of production of antibody is 2000 antibodies or immunoglobulins per second. The total amount of antibodies in plasma protein is 20% by weight. When any foreign material invades our body, then antibody binds to the antigen and activates the various type of white blood cells and complement system. Then they both work together and attack the antigen. The structure of immunoglobulin. Immunoglobulins are typically Y-shaped structure with two antigen binding site. So this is the typical structure of immunoglobulin, and these are the antigen binding site. Means one immunoglobulins can bind to the two different antigenic determinant. Okay, so due to presence of two antigen binding site, the antibody or immunoglobulin is known as bivalent antibody. Okay, so this structure is divided into two fragments, the FAB and FC. FAB antigen binding fragment and FC means crystallizable fragment, antigen binding fragment which bind to the antigen. Okay, so look at this structure. This is the typical structure of antibody molecules. Here we can see that there are the antibody immunoglobulins is made up of 4 polypeptide chain. Among the 4 polypeptide chain, 2 polypeptide chains are heavy. Red color chains are the heavy chains and the blue color chains are the light chains. And these 4 polypeptide chains are held together by non-covalent and covalent bond. Covalent bond, specifically disulfide bond. Here this portion is known as antigen binding site. Here both the light chain and heavy chains are involved. They work together and form the antigen binding site, while the tail region is made up of only heavy chain. Okay, why these chains are known as heavy and light chain? So the heavy chains, or first of all the light chains are made up of 220 amino acids, while the heavy chains are made up of 440 amino acids. Means heavy chains are double in the in their weight. So these are called as light and heavy chain. If we see the types of light and heavy chain, there are two types of heavy chain may found in the immunoglobulins, kappa or gamma, while in case of heavy chain there are 5 different types of heavy chain: mu, delta, gamma, alpha and epsilon. Due to the presence of these 5 types of heavy chain, the immunoglobulins are known as IgM, IgD, IgG, IgA and IgE. So there are 5 different types of immunoglobulin depending upon the presence of heavy chain, what kind of heavy chain is present. So if we see the detailed structure of the immunoglobulin, we can see the polypeptide chains are mainly divided into two regions, variable region and constant region. Here V for variable and C for constant. So this is variable heavy chain, variable light chain, constant heavy chain, constant light chain. Okay, same is here. And these four chains are joined together with the disulfide bond. Okay, as we know that polypeptide chain has two terminal, amino terminal and carboxyl terminal. Okay, and this is the hinge region. It plays a crucial role in the immune system. Okay, so there are billions of different forms of immunoglobulins due to the presence of different amino acid sequence and different antigen binding site. Okay, second is the hinge region. Okay, we see that a hinge region, two disulfide bonds are present here. This is hinge region. Okay, so what is the importance of it? Hinge region is very flexible and it determines the efficiency of antigen binding capacity of the immunoglobulin and cross linking. Okay, so the distance between two antigen binding site may vary, as we can see here. This is antigen and the antigenic determinants are present. Antigenic determinants means the structure which determines the antigenicity of the antigen, and our antibody immunoglobulin bind to the antigenic determinant. Here it can bind to the two different antigenic determinant of the same antigen, or it can bind to the antigenic determinant of the two different antigen. Here we can see that the distance between two antigen binding site, it may be vary due to the flexibility of the hinge region. Okay, and the last point is the tail region. What is the importance of the tail region? Different tail regions are present in different antibodies. Then what is the function of these different tail region? It gives the ability to activate the complement system in some immunoglobulins. In some immunoglobulins, it helps to bind to the phagocytic cell, and in some it may help to cross the placenta from mother to fetus. Okay, among the five different types of immunoglobulin, the IgG antibody immunoglobulin can cross the placenta, means it can travel from the mother to its fetus. So this is about the typical structure of immunoglobulins. Thank you.